So in this chapter, we're going to talk about kind of the basics of the central nervous system, neurons, and their partners, glia. So those are the two major components that you're going to find in the central nervous system, the glia and the neurons. The glia usually inflate, support, nourish neurons. They do lots of maintenance issues. They do a lot of other things too, which you'll explore through some of your projects, some of your research into new ways that glia are being studied. Neurons are considered the main brain function and your main cognitive things where they're going to process information, sense environmental changes, communicate changes other neurons, and command your body responses. I'm going to dictate all that. Over here on the right, you'll basically see a lot of different staining. Is it immunohistochemistry or maybe GFP labeled cells, which I think you did last year in AP Bio. So you can see all sorts of different types of neurons, different shapes of neurons. We'll get into that a little bit more coming up. So first off, here we look at kind of a chunk out of a brain. This is a cartoon obviously right here, but again, we can see our neurons here, okay? They have lots of different processes coming off them, okay? And then they have one process coming down here. This is that thing called the axon. So here, this is our axon coming off. That axon is the major output for the neuron. We'll get into that a little bit more later. But what you're also going to see here are types of glial cells that are providing support. Here we have the oligodendrocytes right here. See, they're actually wrapping around the axons. We'll get into that a little bit, what that's called. So that's basically, it's called a myelin sheath. So you can see here they call this the myelinated axon and the myelin sheath. You also have little things called microglia up here. They're kind of like the cleanup for the central nervous system in the brain. They're kind of floating around, cleaning up all the debris and remains that don't need to be there anymore. And then you see here you have astrocytes, which are kind of all over the place. They're touching neuronal cell bodies. They're hitting on dendrites. They're hitting on axons. And they're also interacting with the capillaries. So they're going to regulate kind of nutrients getting into the neurons. Since there actually is a barrier between the brain and the blood called the blood-brain barrier, something's got to be able to take nutrients into these neurons. So they're very energy heavy. They use a ton of energy. So they need a source for that energy. And if there's a blood-brain barrier where they can't get their energy from directly, well, we've got to get that across through the astrocytes. So they're going to help regulate a lot of things going on with that. So how the brain first gets studied was kind of the basic ideas. Well, first off, we have the Neuron Doctrine. They're saying that, well, there's neurons inside the brain. It's not just some random system of stuff. And this was found through what's called the Nissl stain. Basically, it stains the endoplasmic reticulum with a kind of a violet color. It's developed in the late 19th century. And here you can see that on the exterior portions of the brain, the outermost regions, you're getting a lot of staining. Whereas in this portion, you're not getting that. So that's telling you here we have no ribosomes. And here we have lots of ribosomes. Here we have lots. So that's telling you that you've got a lot more proteins being created in these outer regions. And what we end up finding and seeing is that that outer region is very densely packed with cell bodies. Whereas that inner region that's kind of white there, that's a lot of the axon. That's a lot of the projections where there aren't any ribosomes and no proteins being made. So going back to here, again, here, cell bodies, here, these areas all throughout, those are axons and no proteins being developed. Now, there was a Golgi stain developed by Camillo Golgi back in the 1800s that showed that there's kind of two major parts. Now, what the Golgi stain does is it kind of randomly stains different cells. So normally if you stain every single neuron in a brain, you wouldn't be able to see anything. It would just be so full. But the Golgi stain, I think it is about 9 or 10% of cells, but don't quote me on that one. You can check out that number. And what you actually see is different cell bodies, and you can clearly see lots of different types of um, inputs and outputs coming out of these cells, or things that look like they might be input outputs. So he theorized that these cells were talking to each other through these kind of, here you can see these like dense branches in the cell right here. Here we have these branches for this cell spread out all really wide. So it's probably taking lots of lateral information in, bringing it in, and then it might have this single output down here at the bottom. So we saw all of this and got into thinking, okay, well, the nervous system is probably working by taking information into one location and processing it, sending it out to someplace else, so we can talk to other things that are taking information from other locations. And here's some other examples of Golgi stains. So here we can see a couple different cell types, and you can see a couple differences here too. If you look at the cell on the right here, it's very bumpy on the dendrites, and that's called a spiny neuron, okay? This one on the left is also probably a spiny neuron. Whereas here, it looks like the dendrites are a little flatter in this neuron down here. So that's called an aspiny neuron. Another way of just classifying different types of neurons, and we'll see some more of those classifications in a second. So there were two major people at work. You had Golgi and Cahal, who thought about the nervous system in different ways. Golgi thought that the neurons were fused into sort of a large system, kind of like the vascular, the blood system, the circulatory system, where everything's connected, so everything would just kind of pass from one thing to the other. Whereas Cahal thought there was some kind of circuitry there. Neurons didn't communicate by continuity. There was a contact. There had to be a gap between them, which to him made sense because if you're having all these points coming together, if they were all continuous, it wouldn't know how to decipher kind of what's going on there. So the cells would all remain separate, and they'd act as big integrators of information and then processing out to other cells to integrate that information. It turned out Cahal was right when they really went in and did a lot of electron microscopy. They could see those cells differentiate from one another. So that's what was confirmed back in the 1950s, that Cahal was right and Golgi was incorrect. There is a gap between those cells. So kind of getting quickly, prototypical neuron, of course you have your nucleus where all your gene expression, transcription, RNA processing takes place. All that stuff's going to get processed out to the rest of the cell, but it's all being coded for there, the different types of neurotransmitter that neuron might make, the different types of receptor that neuron is going to express, all that's coded for in the nucleus. Process there through the DNA, through creating RNA, and then that RNA will head out. And just another picture of a neuron here. That RNA will head out into the soma, and that's where a lot of the protein synthesis is going to take place. There's actually no protein synthesis that takes place in the axon. Okay, all of it's going to take place in the soma. There might be some more localized in the dendrites, where they're replacing receptors and things like that, but most of it's going to take place in the soma, and then it'll get transported around. So two different places you're going to have it happen. Some we're going to have on free ribosomes. You notice here we've got a newly created protein, but it's not packaged with a membrane. So this is a protein that's going to float around. So this is just going to float around in the cytoplasm. So it'll modulate some things, it could regulate some different things, but it's probably not going to insert itself in a membrane, whereas this one on the rough endoplasmic reticulum does get synthesized and associated with a membrane. So now it's going to become a membrane-associated protein, which is really important because a lot of the receptors that we're going to see later on as we go through the nervous system are going to be membrane-bound things, whether they're channels or whether they are receptors. Okay, they're all going to be membrane-bound proteins, so they're going to be created at that rough endoplasmic reticulum. And again, just a quick reminder, after we leave that rough endoplasmic reticulum, we go through the smooth ER and the Golgi apparatus, and that's what packages it to be sent so it can integrate into the membrane of the cell. Mitochondria are extremely important because, again, neurons are very energy dependent. So there's a lot of mitochondria in the soma, but there's also a lot when you'll see when we look at some of the um, electron microscopy, there's a lot of mitochondria floating around in that axonal terminal. And that's because this whole process of sending information from one cell to another is very energy dependent. Your brain burns off more energy than almost any other part of your body throughout the course of the day. It's constantly working. Those neurons are constantly needing energy to package off the neurotransmitters to the next cell in the process. Um, so again, neuronal membrane, what's important about the membrane? Well, again, we've got phospholipid bilayer, right? We've got our polar head groups here on top. We've got our non-polar fatty acid chains kind of hanging out in the middle. Okay, super important because that's going to keep things from flowing across. But in the neurons, we're going to have things like this. We're going to have our membranes that insert themselves, and they're going to have regions like this one right here that are going to create pores. And those pores can open and close based on different properties of the membrane, different things that might interact with either extracellular, intracellular sides of that protein, okay, and allow different ions to flow through. Okay. You're also going to have G protein coupled receptors, meaning that if something comes in and binds to this, it's going to activate something down here and do something on the inside of the cell. I'm not going to say it's going to do something specific because it could be any number of things that's going to happen. But different neurons are going to possess different ones of the different types of membrane-bound proteins, either G protein or non-G protein coupled receptors. So there's lots of different types of proteins that are going to be inserted into the membranes. We'll talk some more about those as we get into specific regions. Cytoskeleton, it's always moving around, of course. Okay. Microtubules, microfilaments, neurofilaments, three very important parts. The idea here, though, is that those three tubules here are what's going to carry the proteins. The proteins that get made here have to make their way down the axon, and those microtubules are going to carry them down there. Since none of the protein takes place in the axon, those microtubules are going to be extremely important to make sure that that protein can make it from the soma out to the axon. So the axon has three parts. Okay, has the axon hillock, the axon proper, and the axon terminal. Okay, the axon hillock is the point of what's called, it's going to be where action potentials are generated. We'll get more into what an action potential is later. That is the point where basically an all or none reaction to say, do we have an action potential or not? And then it'll propagate its way. If we do get one, it'll propagate its way down the axon and through the axon proper, which goes everywhere. And then the axon terminal is the point where that axon gets close to another cell. Okay? Or sometimes those terminals actually circle back onto the same cell, but we'll get to more. Again, we'll talk more about that communication later, but just understand those three components of the axon. Again, the ER does not extend into the axon, and there are different proteins that are going to be expressed in the axon than in other parts of the neuron. Now, in the terminal, okay, you aren't going to find any of those microtubules stop. There's going to be synaptic vesicles. So when we see a picture of an axon terminal in a electron micrograph, okay, we're going to see lots of little circles everywhere. Okay, those are going to be our synaptic vesicles. So there's lots of those. You're also going to see when we look closely. We'll get to some later when we get to some other parts of studying synaptic transmission, some real dense dark places. Those are called the synaptic, those are synaptic densities. Okay, basically that's the point where those vesicles will fuse and release their neurotransmitter to the next cell. Again, you also have a large number of mitochondria, okay, here. Okay, again, we need a lot of mitochondria because, again, we're very energy dependent. Okay, synapse. We have synaptic transmission. We're in a, basically there's a point where electrical signaling turns to chemical signaling and then back to electrical signaling. So electricity in the form of an action potential makes its way down towards the end. It causes, that electricity causes these vesicles to bind with the membrane and release their neurotransmitter, which will bind to the postsynaptic site, okay, which may, like I said, could be it'll be another cell, could be an axon of another cell, could be a dendrite of another cell, could be the soma of another cell, but it's going to get really close to that and then release that neurotransmitter to modulate something that happens on that cell. And there's lots of different types of modulation that can happen based on what that neurotransmitter is and what's being expressed on that postsynaptic cell. So there's two parts that are going to affect this transmission, and that's what is in here, what's in here, and that presynaptic terminal, okay, what neurotransmitters are being expressed, and also over here on that postsynaptic side, what receptors are being expressed there. We'll get into more details later. So we sometimes will move vesicles back up. So we have intrograde. Again, that's bringing everything from the soma down. Okay, retrograde is taking it back. Again, that's where we might repackage vesicles or recreate proteins. Okay, that way we can restock our neurotransmitter supply. So the dendrites, that's where the incoming signal comes from typically. Okay, so this right here is called a Purkinje cell. Purkinje cell. They're very cool. They're in the cerebellum, and you can see this massive dendritic branching up here. Okay, and the thing that's really cool is if we turn this thing sideways and looked at it kind of on from the side, it would basically just look like a stick, like this, because it's super thin. The tree doesn't go, it's very two-dimensional, it's not actually three-dimensional. So this is where our incoming signal is mostly going to come from. It's kind of like the reception zone, and you're going to get signal coming into all these different branches, and it'll make its way down to the soma, and that's where the decision will start to happen about what's going to take place in that neuron. How is that signal that's coming in from all the other cells that are touching it affecting the output of this cell? So a couple different classifications really quick. We have a single neurite, which means it's unipolar. Okay, so here's our unipolar cell, this first one. Okay, we have two neurites here, which is a bipolar cell. I'm not going to bother writing, it's labeled right there. Or we have multipolar cells. Most of our neurons are going to be these multipolar cells, but there will be some unipolar cells, and there will be some bipolar cells that we'll interact with a little bit. Other ways of classification, we have some stellate cells, which are more star-shaped, like this one on the right. Okay, we also have a thing called pyramidal cells, where their cell body kind of looks pyramidal. So that cell body right there kind of looks like a pyramid. They're all throughout cortex. There's tons of them. So in your cortex, in any animal that has been looked at, they really provide a lot of the major processing layers in the cortex. Some further classifications is by their connection. So are they primary sensory neurons? Are they the ones that get the initial input? Are they motor neurons? Do they connect to motor outputs? Are they interneurons, meaning do they connect between other neurons? There might be some things based on axonal length. How far does their axon stretch? One of the big types of classifications is neurotransmitter or maybe also receptor type. So you might have cholinergic neurons, which means acetylcholine is produced at their synapse. You have GABAergic neurons, meaning GABA is the neurotransmitter that's being produced. So that's a good way of classifying a lot of different types of neurons you'll hear me talk about as we go through everything. So glia, what are they? Again, they're going to support neuronal functions. Astrocytes are all over the place. They fill in this space. They absorb neurotransmitter. They influence what happens with the growth of new neurons as they're growing. They regulate the content of the extracellular space, like what is around all the different neurons. Then you also have myelinating glia. These are the oligodendrocytes or the Schwann cells. The oligodendrocytes are what's in the central nervous system. The Schwann cells are in the peripheral, meaning outside the spinal cord. So they basically insulate axons. So in here, this area, sorry, let's go. So this area from here to here is the axon, the tube. Okay, you can see some microtubules kind of sticking through it. And this like onion-y type layer section right here, those are the oligodendrocytes wrapping themselves around that axon bundle. And you can see a lot of those different ones right here. Now those wraps, you've seen this in the cartoon, have gaps in them. There's a break here called the node of Ranvier. Okay, the node of Ranvier. Basically, what that wrapping does is allow for the signal to transmit a lot faster. And this node of Ranvier is basically where the signal gets, the signal strength is increased. So the membrane's exposed, we can reestablish the action potential, okay, because it might have leaked out a little bit as it was passing down the axon, but here we're gonna increase that strength again, get it back up to where it was when it first started, and really allow for that signal to pass further and faster. Our last one for glial cells are called microglia. They're kind of the phagocytes, meaning they're the immune cells that are going to destroy things. You kind of see them here in green throughout this tissue sample right here. Okay, they're basically going around cleaning things up. They're getting rid of old neurons that have died, maybe old synapses that broke down. They're cleaning that up. They're absorbing things that aren't supposed to be there. They just walk around making sure things are preserved correctly. So that tells us a lot about kind of the basics of everything that's part of the nervous system. And we'll go into how all of those things start to communicate throughout the entire course.